Thank you so much, Roland, for this nice introduction. And I would like to say thank you for the invitation to this workshop. I'm really excited to be here. I find it so inspiring, especially also the interdisciplinary setting, including the interdisciplinary audience. And it is very nice. And I'm really glad that you are still all here. Very nice. So I hope I will not disappoint you by moving on to organisms now. And as Roland said earlier in the introduction for this session, by looking at the organisms, we look back, right, if we are interested in evolution. And this is another chance to approach, you know, the important steps in life's evolution, right? And I will touch this a little bit today. So yes, I have been interested in archaea for a long time, and to tell you what archaea are, you might be wondering. Archaea are microorganisms. They are as small and as simply built as bacteria, which means that from their shape, they are rather boring organisms, if you want. But if you look into what they are able to do, their metabolisms, the conversions they can do, the geochemical conversions on Earth, and also their molecular biology in general, signal transduction and so on, is amazing. So there's a huge diversity in the archaea group, but also in the classical bacteria, yeah, which is the second primary group, if you want, on Earth. We will come to this. So yes, archaea have a classical microbial cell structure, if you want, cell shape, most of them. And archaea have a specialty that they share a taste, or many of them share a taste for extreme environments. So I have also been on volcanoes and in hot places. This is what brought me to the field. I did my PhD with Wolfram Sillig, and he was an adventurer, also bringing hot organisms in the 1980s, and then with me also in the 1990s. This is me on Mount Yo in Japan, and it's probably the last time that I followed him on such extreme places, actually, but this was very exciting. So this is one idea about archaea, that they are specifically adapted to extreme environments. And it's true that, especially with respect to temperature, this is where they have diversified. In hot springs, they have diversified. We find most lineages and metabolisms in hot springs, although they are also occurring in our gut and on our skin, and also in moderate environments. They also define some physical borders of life, if you want, where we know that life exists. So the hottest temperature that is known where organisms are still dividing is currently at 122, and this is an archaeon. And I have to name Karl Stetter here, also a member of the Leopoldina, who has isolated a lot of organisms. And I think he made it up to 121. There was this fight about, because 121 degrees Celsius is the temperature of the autoclave, right, where we sterilize normally. So this is around that edge where we find organisms that can still divide. And from this trip to Japan, actually, we isolated Picrophilus oshime, which back then had an optimum of, it still has this pH optimum of 0.7, the lowest back then, I think maybe still, and it grows at pHs that are negative even. I didn't even know before that this exists, actually, or it could. But I would also like to mention that archaea play an important role in the ecology of the planet in moderate environments. So they are also very important in the nitrogen cycle, and it was actually my group who discovered them in large abundance in the soil, performing the step of ammonia oxidation as part of nitrification. You might know that this nitrogen cycle is solely performed by microorganisms, right? And so with the reactive nitrogen species. And it's a very important step. And you might also know that we have the problem of over-fertilization. So a large part of my group in the last 20 years has focused on the problem of nitrogen and the involvement of archaea in this, which also involves as a side product N2O, lachgas, which is also produced by denitrifiers, but also in the nitrification process, which is, of course, a problem, and as is methane. And you might know the methanogenic archaea from another member of Leopoldina, Rolf Tauer, who has done seminal work on the methanogenesis and on the metabolism. So these are important roles of archaea as well, to cover this part in the ecology, and now I will come to the evolutionary aspects. So I think that most of the scientists' view converge to the fact that there was a very early split among the microorganisms on Earth into bacteria and archaea. And this split was found end of the 1970s by Carl Woese, who was the first to do sequence analysis with ribosomal RNA, a conserved molecule, to actually find a natural phylogeny of the organisms. And he made this seminal and very astonishing discovery that there is apparently a deep split between the microorganisms. And this finding was supported over the years and decades by many different studies. One of the early supports for this, apart from sequence analysis, was the study of their lipids. And maybe you know about this, so I will cover a little bit of the lipids here. Maybe you know about this. The bacteria and also all plants and animals, all higher complex organisms, have ester lipids linked to glycerol, and the side chains are fatty acids, and they form a bilayer. And opposite to that, only archaea, and all archaea, have ether-linked lipids to the glycerol. The glycerol has a different stereochemistry, and the side chains are not fatty acids, but actually isoprenoid side chains, so branched side chains with different modifications. Plus, we can have monolayers that are linked in the middle. And this makes them predestined to live in extreme environments, makes a lot of sense. But these lipids are also found in the archaea that we have on the skin and in the gut and in the oceans. So it is not per se an adaptation to hot temperature, but certainly a good adaptation to be able to sustain. Another important aspect, apart from different cell membranes and also cell walls, by the way, that was Otto Kandler, also in Germany, strong German input actually on the archaea field. Otto Kandler found different cell walls in the archaea. This is also supporting this, but there are big diversities in the different cell walls. So this is not like a unique feature like membranes. But another very specific feature or characteristic of archaea is that they have molecules in their information processing that are more complex than in bacteria. So here you have the example that was discovered first and is most famous. It's the RNA polymerase that reads out our genetic material from DNA into RNA. So here we go the other way around if you want, when you go, think of the RNA world. So the RNA molecule in the common organisms is the intermediate between DNA and proteins. And so this is the central molecule to do this. And the RNA polymerase, interestingly, of archaea is more complex. You see this homologous subunits. Here's the crystal structure nowadays available. The homologous subunits are colored in the same way to show you this. But in the next slide, I show you what the RNA polymerase of eukaryotes looks like, so of plants and animals and us. So this RNA polymerase is very similar. And this is a molecular tree, if you want, a tree made out of sequences also, where you, which represents also what Carl Woese showed. Maybe it's almost out. So that you have a common ancestry. Isn't there another pointer somewhere here? Yes. You have a common ancestry between archaea and eukaryotes, and it is explainable that in this time here, a common mechanism evolved to transcribe DNA. But the same is also true for replication. So the way the organisms duplicate their genetic material before they divide is actually also quite conserved among archaea and eukaryotes. So this is quite astonishing that these small organisms with their small genomes have certain machineries that are as complex as those in the higher organisms. This was a huge surprise, actually, and really supports this earlier linear phylogeny. So to sum this up and to wake you up, I use a little metaphor, if you want. If you think about it, what it really means is that if we look at information processing molecules, then this organism, Pyrococcus furiosus, growing at 100 degrees, dividing every 40 minutes, it's one of the fastest, is less related to the bacteria like E. coli, 37 degrees, that divides every 15 to 20 minutes, but actually it's rather more closely related to, for example, the president of the Leopoldina. And this is also true, is he still here? This is also true for replication, as I said, for DNA repair molecules, to splicing. So a lot of events that happen in the eukaryotic nucleus, right? So, and this brings me to the transition and to the actual work that I would like to share with you, that is a new finding in the evolution of eukaryotes. And I dare to ask the big question, one of the biggest questions maybe in biology, which is, how did it actually happen that from the simple organisms of prokaryotes that we come to the complex organisms, multicellular organisms, and so on. This lies, of course, in the moment when the cells changed, right? So we have prokaryotes, archaea, and bacteria, and then we have the eukaryotes, which is single-cell organisms first, most probably, and then multicellular organisms of all the complex life forms, plants, animals, fungi, protists, and so on. And these organisms are called prokaryotes. Maybe you remember this. Prokaryon means before the nucleus, because the biggest distinction between these two cell types, apart from their size also, is the nucleus. So the genetic material is inside a nucleus, one compartment, and then there is more compartments, and the cell is relatively complex. Here are the mitochondria, you have Golgi vesicles, and you have this endoplasmatic reticulum, which is like the way molecules are transported and so on. So we have this extensive compartmentalization in eukaryotic cells. And one of the most famous hypotheses that I think is not a hypothesis anymore is that bacteria, the bacterial side, actually brought in the mitochondria. And this hypothesis has been out for a long time. And I think it's not debated anymore, really, because any organism that is being sequenced and where the mitochondrial chromosome, what is left over of it, is analyzed, shows that, that these can actually be traced back to a proteobacterial lineage, probably of the alpha proteobacteria. So this is pretty clear, but it was always unclear which organism actually engulfed or mated with the bacterium that gave rise actually to the eukaryotes. So this is actually the view that I think represents better the organismic tree of life, where you don't think linearly that the eukaryotes come out of archaea somehow, but we rather have to also consider endosymbiosis, for example. So Carl Woese, in the end of the 1970s already, and all his life, was thinking about what it means to find another primary lineage here, and how actually this eukaryotic, the eukaryote evolved. And he suggested, he was one of the first, I think, to suggest that there was a proto-eukaryote, a cell on the stem, where we also find archaea, that evolved a nucleus, however it did this, and then was able to engulf the mitochondrion to form the eukaryotes. And so genome sequencing over the past years has shown that we cannot find such an organism. There are some organisms, protists, flagellates, Giardia lamblia, for example, that don't have mitochondria, but we find remnants of the chromosome of mitochondria in the host's genome. And so there's not a single organism found that actually was in the time before this endosymbiosis. So the alternative hypothesis that emerged more and more also from phylogenetic studies was that there is a lineage of archaea, because archaea have developed all these information processing systems. There would be wonderful hosts actually to take in a bacterium, and maybe it was this moment of an archaeon and a bacterium 2 billion years after they emerged first on Earth, that they found each other again and actually formed the eukaryote. And this is what I'm going to share now with you. In a few minutes, you will know what this lineage might have looked like, because maybe it was found. And yeah, and this is what you will hear about now. So the story actually began in the northern Atlantic with the discovery of hot vents north of Iceland by the Norwegian geoscientists, who were looking for the northernmost hot vents, and they discovered what they called Loki's castle. So they named it after a Norwegian god that likes to change its shape, and this is what hot what black smokers, for example, also do. So these are some pictures of this place. And back then, I was a professor in Norway, and we had the task to look a little bit into the microbiome, but actually of the marine sediments that were further away, because it was clear that the plume down there in the ocean, this hot vent is about 2,300 meters deep, that it might have an effect on these sediments that are forming there. They are extensive sediments. And this is a real X-ray picture. This is what I mostly want to show you. So it is actually highly stratified. There is fallout from the plume of these hot vents, although we are a kilometer or so away. This is like a real photograph, if you want. This is a 3-meter core. And so because of this unusual stratification, we were able to find stratification also in the chemistry and also in the microorganisms, more than usual. So we were able to find layers, although this was not at fine resolution yet in the first study, but we were able to find layers where we have an unusual augmentation of certain groups of microorganisms. So what we did is we isolate nucleic acids and we do some sequencing of conserved ribosomal RNA, just like Carl Voss, and then we find sequences that we can assign to, back then, completely unknown organisms. They were named the deep sea archaeal group, for example, DSAC here. Nothing was known about these organisms, just a snippet of their ribosomal RNA. And because we found them so highly enriched, like up to 50% of the microbes in that layer, we were able to do some genomic analysis, which happened years later, in collaboration with 2 working groups. And indeed, it turned out that these genome pieces that could be assembled to almost full genomes, this is metagenomics, the new era of microbiology, if you want, indeed, they were the closest relatives when you did phylogenetic study with different genes, as it is done. They were the closest found relatives to eukaryotes, to plants, animals, and other genes, yeah, from the complex organisms. And the other gray lineages here are actually other archaea, and then you have bacteria. So they seem to be like the closest sister group of eukaryotes. And this has triggered a lot of more studies. So people went out to all their samples in the refrigerators, including myself. We were looking for other DNA if we can find them somewhere else, because once you have a sequence, you know what to look for, then you can have a lot of resources. And genome sequencing nowadays is an easy task. So it was an explosion actually in the field. A lot of people jumped on it. This was a big work also of Thijs Ettema's group, who got really famous with that because he is doing a lot of this genomic analysis and assigning genes and so on. So now we have actually 12 Norwegian gods, 12 lineages that are named after Norwegian gods like Thor Archaeota, Odin Archaeota. It all started because of Loki, Hel Archaeota, and so on. And now we also have actually, since recently, a Chinese god that is added to this. But the naming of the Asgard actually stemmed from the fact that there were all these different lineages found, which means that this group of organisms has a huge diversity, and more lineages can be found. And then a lot of phylogenies were done, and I've discussed this already earlier today, so the phylogeny is not always as clear that we have like Loki, Archaeota, or others being close to eukaryotes as this sister group, but I would say like in 60% or so of the cases. And interestingly also, there are genes in these genomes that made it even more convincing. There are genes in those genomes that you would not expect for a prokaryote, for a simple organism. And these genes, for example, I just point out what is relevant for the talk, are genes involved in membrane remodeling, which you have found a little bit maybe before in bacteria, but in all these Asgard archaea, which they are called now because that's the home of the Nordic gods, you find a lot of them. And then also there were genes for cytoskeleton, for actin, which you usually only find in eukaryotes, and even proteins that make the actin dynamic. And the actin cytoskeleton is what we need for our immune cells, for the macrophages, what we need in our nerve cells, what is in every eukaryotic cell and essential and very important. And the hypothesis was, does this organism, this archaeon, already have a cytoskeleton, which would be completely unexpected, right? So there were a lot of predictions, a lot of hypotheses, and people went out to culture, including also my group. And our sampling site was first the Danube in Vienna, but we did not find enough organisms. So we went out actually to Piran in Slovenia, which is only 5 hours drive from Vienna. And I tend to bring young students there. And I realized in retrospect that this is a good way of getting young scientists addicted to cultivation also, because one has to have a lot of endurance actually to cultivate organisms that are hard to get. So this is how I get them, I realized. And yeah, and at the end, the successful cultivation that we had took about 2.5 years, but in fact, it was a 6-year endeavor, of which the first 4 years or so were not successful. And this is what the data look like when you look into your enrichments, you get all these signatures of very many different phyla. This is the complexity in the sediment, just by colors, the different phyla of organisms. We are talking of thousands of different sequences. We make 15,000 to be kind of representative per sample. And these are just the bigger taxon levels that you see here. And then you see how we become more and more, you know, how we get closer to enriching for Lokiarchaeote. And then came the interesting aspect. Now we are going to look at them. And that was very exciting. So here we have an enrichment, so it takes a month until we have a culture ready. So it's not like an overnight bacterial or archaeal culture. It takes 2 months actually to get them ready to grow. And but what is really rewarding is that they have a very unusual shape, very unusual. Nobody had predicted it actually until our colleagues from Japan had cultivated them. So ours is the second cultivated organism. The first one is this one here, Prometheaceum syntrophicum from the Japanese group, Imachi et al., which was published 2 years before, and is similarly complex. So these organisms are very unusual for prokaryotes. They have very long protrusions, and yeah, and they show that there is maybe a different way of complexity. So we were on conferences when we had the genomes first, and we were debating what these cells might look like, and we expected a lot of complexity inside the cells. But in fact, they actually show this to the outside. And here you see, this is nice. So the cells are very sensitive because of these long arms. This is what made the Endivia really hard to cultivate them. And what is the advantage is that they are very small and that you can put them into one of those novel machines in biology, which is cryo-electron microscopy, and you can look inside. And so this is pictures of a putative organism from the distance first, from like transmission electron microscopy. This is a bacterium inside the culture. This is another archaeon. And you see here the different cell walls. So the bacterium has a double membrane. The archaeon has a beautiful S layer on top of the membrane. And this is what these organisms look like. We called the organism Lokiarchaeum ossiferum. I will explain the name in a minute, but it has a very unusual cell envelope, if you want, because it doesn't have really a cell wall. And then when we can identify the organisms, it's a longer way to go, and this is published. But then when we were clear which organisms these are, we found a lot of interesting structures, and inside we see, most importantly, we indeed see cytoskeleton. And this is a cell body, and here you see some protrusions, and I show it a little bit in more detail on the arms. So these were protrusions, and I want to give you a glimpse through this, you know, like scanning through in the depth. So you see that there's a lot of those filaments inside the arms, and you also see ribosomes, which are the molecules. It comes back. Which are the molecules that actually make protein. So we see that inside these arms there's apparently protein synthesis happening, and you see a lot of actins. Oh, sorry, I'm too far away, sorry. So you see ribosomes and cytoskeleton. It goes on, but I was waiting for a coloring for you. Here it goes. So this is what we see. In the protrusions, we have actin filaments, and we have ribosomes. And what we don't have is actually DNA. When we stain, I've taken out those pictures for the sake of time, but when we stain the cells, we see really a cell body, and then we see these long protrusions, which apparently have something like cytoplasm, but they don't have DNA. So we have the feeling that there is some sort of pre-compartmentalization that is usually not found in prokaryotes. And for sure, what we have is actin, Actin is the molecule, the basis for cytoskeletons in the eukaryotes. And yeah, there are some more extra interesting features on the outside of the cells. But what I want to show here is, and here we go back, for example, in time. So what I want to show you is that this type of actin, cytoskeleton protein, is found in all of these different lineages of the Asgard archaea, and it is closely related to the bona fide actin of eukaryotes. So we think that at the beginning of when Asgard split off from other archaea, that this actin must have been in place, and it's the most highly transcribed, but it's also one that we were able to stain. So yeah, so this is very interesting, and now we are going, of course, further to study this cytoskeleton. Another feature, and that's already the end of my talk approaching, another feature of these organisms is that they have this sticky way of growing with other organisms, probably because of their long protrusions. So they enclose also other organisms. These are methanogenic archaea in the culture, in this picture, for example. They grow in very complex structures. It's very difficult to transfer them, to quantify them, to stain them. It's not easy to, so it's a new microbiology if you want, but they have very interesting features. And this is already the end of my talk, so I would like to close the circle here and show you what the current opinion is in the field, actually. So we think that Asgard archaea, by phylogeny and also by their specific features, support the idea that there was this lineage of Asgard archaea that have played an important role in the rise of eukaryotes. And there are many more features now that can be studied because we can culture them. There are also very nice hypotheses how this might have happened, how this eukaryogenesis might have happened in the moment of endosymbiosis. But of course, we have to predict many more things. We have to predict that the first eukaryote actually had first a mitochondrium and then developed the nucleus, having two genomes here, two different types of lipids that it had to deal with. A lot of things must have happened here until you actually come to a eukaryotic cell as we know it today. But of course, these predictions are needed to follow up in models. So I hope I can share my excitement that there's a new field now of research actually coming. There were decades of research on archaea, on all these information processing systems that have a common evolutionary origin with eukaryotes. And now all of a sudden there's a novel lineage that is even closer to it, which is really exciting for archaea research. And with that, I would like to thank my people, those, particularly those that were involved in the study, Thiago, who did all the cultivation at the end, and Raphael, the bioinformatician, and then the people who have helped in cultivation over many years. And I must say I was glad to have long-term funding, and if there hadn't been COVID, I would have not had any results after 5 years with an ERC grant, which would have been embarrassing almost. So it is good to have long-term fundings, I must say. And the amazing electron microscopy, the cryo-EM was done in Martin Pillhofer's group, mostly by Florian Wollweber. And with that, I would like to conclude and thank my group, actually, amazing people, many of them willing to cultivate difficult to culture organisms. Thank you.